also see you. The results that I'm showing today are part of a project which is termed START, which stands for the Singapore Techno Alliance for Research and Technology. And as you can gather from the name, this is a joint collaborative effort bringing together 3 different institutions, the NTU and NUS over in Singapore, and the Technion over the State of Israel. And this is funded by the National Research Foundation of Singapore. And the idea was to put together an interdisciplinary team of clinicians, cardiologists, basic scientists, applied scientists, with the aim of providing regenerative medicine solution, intermediate and long-term solution for clinicians dealing with a failing heart. And naturally, due to the nature of this conference, I will be addressing the biophysical aspects of cellularization, and I would like to share with you some I believe, an unorthodox approach we are taking in START. And I would like to have a fruitful discussion with you. And I mean, one of the things I love about this conference is the fact that there is really good discussion here happening. And I think since we've been trying to develop a particular approach, I would appreciate to get any feedback from you, what your thoughts are on this matter. So I think the subject of the ECM and the relevancy will become clearer as I advance throughout this presentation. Just in a nutshell, I mean, most of you are probably familiar with the concepts of tissue engineering and this pyramid involving the 3 different components that are required to engineer a tissue. Maybe an emphasis would be on a subfield of tissue engineering, which is called functional tissue engineering, where we're not dealing with just any tissue. We are dealing with tissues that have a specific load-bearing function within our body. And it's not that those parameters that we see here are not important for all tissue types that we're developing, but the emphasis might be a bit different. If we're talking about non-load-bearing tissue, for instance, such as liver, primarily the choice of cells, the top of the pyramid, will be the factor affecting the result that we get, because the liver primarily functions as a biochemical pathway. So we need to address the cells and their ability to provide a function. But if we're talking about bone, for instance, then the choice of scaffold, the design of the scaffold material that we are using, the external stimulation that we are providing, those are the key factors that will affect the success. It's not that the cells are not important, but the primary design criteria would actually be the base of this pyramid that we're looking over there. And so naturally, if we look on review PubMed or we look at the literature, most research is done along this base axis of the pyramid. And if cells are involved, they would usually be towards this direction, as you can see in the arrow. How would the choice of scaffold, the design of the scaffold, how would the external stimulation that we provide, how do those affect the cells, the cell phenotype? And there are a lot of publications along this way. Some of them were mentioned by Lisa earlier today. And they show exactly changes of phenotypes of cells and changes in the differentiation state, et cetera. But as far as biophysics is concerned, or biomechanics, not many are asking the inverse of those questions. Not many are looking into what happens to the scaffold when you put cells in it. How do cells affect the biophysics of the scaffold? Or to put in other words, what do cells look into in the scaffold when they get there in order to try and recreate their niche, recreate a tissue out of it? And this is an approach we've started to take over at START. And of course, we will not be dealing with all the tissues in there. We are specifically oriented around muscle, and not just any muscle, the myocardium. So this is myocardial functional tissue engineering. But I think the concepts I'm presenting today could probably be beneficial to other disciplines as well. So in a nutshell, for those who are not from the cardiovascular system, the damaged tissue that we are trying to regenerate is this scar tissue that you can see here. This is a fibrotic scar tissue that it caused following myocardial infarction. This is a cross-section of the left ventricle. We're looking at it from above. And you can see that depending, of course, on the extent of the damage, this yellow scar tissue could be a transmural damage, the entire thickness of the left ventricular wall. And it can span 10 to 15 cubic centimeters in volume. So it could be quite a large damage. And the problem is that this scar tissue is less resilient than the remaining healthy tissue. It is not active. It puts on extra burden, actually, in the remaining myocardium. It needs to constantly contract because this is a load-bearing organ. And with time, the cyclic stresses and strains that are exerted on this scar tissue will cause it to thin, to become dilated. There are some cellular processes involved called remodeling that have been documented. And the end process of that, the heart becomes enlarged. Now, again, depending on the severity, this could take a few months. This could take 10, 15, 20 years. But the end of that process will be heart failure, which is clinically defined as inability of the heart to pump sufficient blood that meets the body metabolic demands. And unfortunately, current therapeutics, whether surgical or pharmacological, do not really regenerate these scar tissue. So they do a bunch of other stuff, but they don't regenerate that. And that's where myocardial tissue engineering enters into the picture. And of course, this is a load-bearing organ, so our first and primary question would be what sort of scaffold we need to use. And there are various ECM mimicking scaffolds, and I call them ECM mimicking scaffolds because by definition, a scaffold should basically mimic the ECM. I mean, if you look in tissue engineering textbooks, most of them, and I think, Lisa, I've shown it on your slides as well, most of them will define scaffold as having at least those three initial parameters that the ECM is characterized by. The fact that it should be biocompatible, provides 3D architecture and mechanical support, and be hopefully biodegradable if this is a desirable approach for muscle tissue engineering. It is. So these are actually common between ECM materials and scaffolds. But then this fourth point here, which is rarely recapitulated by various scaffold designs. And we also in START, we develop a lot of scaffolds, I mean, synthetic and natural origins, and many other research groups around the world. But it is this point that distinguishes ECM from most of the developed scaffold designs available out there today. And that is the fact that the ECM is not just a static component, or maybe a static component that should be degraded. It's a bioactive dynamic entity. It's been constantly remodeled by the cells. It responds to the pathophysiological conditions in the body. And in response to that changes, it's been remodeled by the cells. In response to those changes, it also affects the cells' regular cellular processes such as migration, survival, differentiation, proliferation, trafficking. All of these have been regulated by ECM. And in the case of the heart, it also modulates contractile force transmission. And these are things that are not easily recapitulated in a scaffold, a synthetic scaffold design. So that's why we have chosen to ask our primary questions on the effect of cells on the scaffold using the ideal scaffold that we can think of, which is the decellularized porcine cardiac ECM, or PCECM for short, as would be abbreviated from now on throughout this presentation. And you may ask why pig? Why porcine? So, the pig anatomy and physiology are quite similar as far as the cardiovascular system is concerned to the human being. I mean, there are those who would argue that perhaps there are other similarities, not just anatomical and physiological, but I mean, looking into this possible, well, I would call evolutionary pathway of mankind, we are harvesting hearts from young and very enthusiastic pig volunteers. And those hearts then go through a process of decellularization, whereby we carefully remove all the immunogenic cellular components from the tissue. So what we are left with is a 98.9% genetically conserved proteoglycans, proteins, glycosaminoglycans that are present in there. Those are bioactive molecules, which are immune tolerated. Actually, they're biocompatible, but I have the data, but I will not show it today. And the first question that we're asking ourselves over at START is, what are the similarities and the differences between this ECM material and the native tissue from which it was derived? Similarities, of course, are very encouraging because this would suggest that this scaffold is ideally suited. It's matching biophysically or biomechanically. It is matching to the heart, so we can implant it. But it is those differences that we are more interested in, because those differences would suggest that we should try and focus on those as sort of an implant design criteria to try and improve our material towards a native state in order to get better implants and better outcomes. And so there are two approaches we can take in trying to deal with those differences. One would be what synthetic material scientists would use to do would be some biochemical modification to the scaffold. And other biologics would tend to do some cellular work, put some cells on it and let the cells remodel the scaffold and see what happens. And we actually took both approaches in the start, but for the sake of today's talk, I will refer to cells as a natural ECM modifiers. And what we are interested in studying in vitro under various static and dynamic culture conditions is what are the effects of cells when they interact with the ECM? How do they affect the biophysical properties of that ECM? But then, I mean, we are using mesenchymal stem cells because this is a remodeling capable cell, and it's a very good cell source for regenerative medicine. So we are studying mesenchymal stem cells, human primary mesenchymal stem cells as a model. But then we're also interested to see what happens when this scaffold is implanted in vivo in an immune-competent animal model, because then we're not just looking at a particular cell type, we are also looking at the entire range of cells that the host can display, including the entire innate and adaptive immunity. And so a while back, a few years, we have already described our decellularization protocol. Basically, you can follow this here in this sequence of images. You can see that if we start also with a sort of carpaccio, like slices of the porcine myocardium, I mean, I'm not recommending eating it, it's just a sort of a description. You can see the process whereby we remove the cells, the brown yellowish part is being removed, and what we get at the end is this insoluble white, presumably due to the vast amounts of collagen, matrix. And you can actually see preserved architecture of blood vessels within this ECM material. And if we do histology, this is Mason trichome staining. So Mason trichome stains blue for for ECM fibers, collagen, and red for cytoplasm of cells, and black for nuclei. So you can actually see that the ECM is completely devoid of cellular materials. Instead, we have the ultrastructure here of the fibers, and you can really see how well nature made it. And so this is indeed an acellular material, and then we can continue on with that. And the first question, as I told you, we were asking ourselves, and we published that a few years back, was how can we compare the native tissue to ECM biomechanics? And we did a number of assays, standard instron uniaxial testing, cyclic strains, stress relaxation, and strain to break. You have a few repetitions and the representative curves, the average curve here in the middle, and then you can see both representative curves for the ECM, the native tissue. ECM here is in blue, native tissue is in green. Also here, this is strain to break. And similarly, you can see here the cyclic strain value. So just by observing those three graphs on the bottom, we can see that there are a lot of similarities in the overall profile, but then there are also some differences. They don't really look exactly the same. And we asked ourselves, can that be quantified? And so the first thing we did was try to quantify a few parameters. And I've pointed out in blue here those similarities. For instance, the energy dissipation is the area blocked within the curve here. Okay, so this area blocked within the cyclic strain assay suggests that they are not significantly different, the native tissue and the ECM. And also the toughness, which is the area blocked here underneath this curve, it's not significantly different across the data set. Meaning basically that suggesting that this material could probably withstand the cyclic stresses and strains in the heart. It could be implanted as is, acellular without cells. But then there are other parameters which are different. All those that appear here in red, they are significantly different between the groups. And so the next question was, can cells restore those biophysical properties towards the native state? And we're thinking of what can we do to prove that. And the first experiment that comes into mind is let's just put cells on the ECM material and see if by handling, okay, we can actually see a difference. And this is one of those few occasions where a hypothesis actually works and you see the differences quite instantly. I mean, I would like to point you out to this curvature that you see over there, which you will never see in an ECM. I mean, the ECM will always take the shape of whatever dish it is in. It sort of collapses. And once you put cells in, if you give them enough time, eventually they will shrink the scaffold and they will give you this sort of appearance, which is similar to the native tissue. And so we asked ourselves, okay, would that be quantifiable? And indeed, it is quantifiable. And for many, let's concentrate on these red parameters, which are different before. So for many of these parameters, we actually get significant difference from the ECM to the reseeded. Also here, the ultimate stress and the train strain. In some occasions, also, it's non-significantly different than the native tissue, actually. But unfortunately, the Young modulus, which is a very important parameter in biomechanics, does not seem to be improving by the effect of the cells. And so it took us a while to understand why. And then one of the folks over at Star said, well, you're doing uniaxial testing, and in the heart doesn't function in a uniaxial manner. The heart functions in a biaxial at least, or three-dimensional stresses. So we're trying to do biaxial mechanical, equibiaxial mechanical testing, which for Young modulus is quite easy, relatively easy to do. And there we actually noticed that the cells have sort of a preferred alignment. If we seed the surface and we look at the surface of the cells with wet SAM, we can actually see the cells aligning on the fiber preferred, the green arrow over here, that's the fiber preferred direction, on what is here marked as a y-axis. Okay, and we can, of course, because this is biaxial, we can test both the fiber preferred and the cross fiber preferred direction, which is the x here. And in both cases, I mean, the y-axis is more significant. We can see basically a reversal. The Young modulus is reduced from the ECM towards the native state by seeding. This is the seeded scaffold, and this is the native. So basically, Young modulus is reduced by cellularization. And the first thought that we've had, how come Young modulus is being reduced by the cells? What do the cells do in there? And so we started looking deeper. By the way, the anisotropic ratio, I mean, we were supposed to have a talk earlier today about anisotropy. So the anisotropic ratios remain the same in all samples. But in order to understand why we have a reduction in the Young modulus, we started looking, sort of doing a longitudinal sectioning and looking with SEM from within, what happens in the bulk of this sample. And if we look at that way, or if we look at on a transverse section along this direction, then we can see the fibers of the cells intertwine between the ECM fibers around them. Here you can see like a 3-cell bundles, and you can see that those thin filaments of ECM fibers that are connected them one another and also to the neighboring ECM. And also this is H&E. You can actually see a collagen fiber at the edge being remodeled. This is an alpha helical structure. You can see it being remodeled by the cells, and the cells are actually, I mean, this is much higher magnification, of course, lower magnification, sorry. But you can see the cell nuclei in there. You can see them really penetrating in between the fibers of the ECM up to 100 micrometer from the surface, which is known diffusion limitation. So what we suggest is happening in there is basically that the cells act as sort of biological plasticizers, I would say. They get in between the fibers, and by getting in between the fibers, what they do is basically pushing them aside, preventing entanglements from happening. And thereby they reduce the Young modulus. This is our working hypothesis at the moment. And, but it got us thinking again that there are two types of parameters that we could probably try to monitor in addition to mechanical properties. And I think that will be my last slide, or my last couple of slides. It's the surface and the bulk that we could monitor as well. And so monitoring the surface, we did a wet SEM and FTIR. You can see the difference between the native tissue and the ECM, which is a very porous structures with fibers. And of course, the seeded, which is the same image as you've seen before, it regains back towards a native tissue appearance. And FTIR, of course, we cannot compare those representative graphs. What we need to do is to normalize the peak intensity ratio to a standard peak. So when you do that, you can actually do some statistical comparison between the groups. And we are left with two opposing phenomena. One of them is strengthening of the amide bonds, which are represented by those wavelengths over there, particularly the 1550, where you can see a strengthening. I mean, if this were the native tissue and this is the ECM, so when we put the cells, the amide bond strength is increased. And what we think is the reason that even though the ECM is made out of proteins, it is very porous. So if you look at FTIR, you're measuring the density of proteins on the surface. And when you put cells in there, the amount of protein that you have on the surfaces generates at the end of the day higher density than what you would have in an ECM, which is a porous structure. And then a complete opposite phenomena as we get with those peaks over here, those peaks which are generating which are sort of marking of acylated glycosaminoglycans location. And those have been reported to be in the ECM, both in our publications and in others. So those acylated glycans are part of the ECM. And when the cells grow on top of it, they basically mask those acylated glycosaminoglycans. So that's why we see this phenomena of the native tissue. This is the ECM, and then when the cells grow, they go back towards the native state. So we see less acylated glycosaminoglycan on top. So cellularization increases the amide bond density and masks acylated glycosaminoglycan on the ECM surface. How is that important? So glycosaminoglycans are an important factor in hydration of scaffolds. And they just generate a lot of hydration around them. And so that's got us thinking whether we can measure in the bulk, for instance, the water content. So we used thermal galvanometric analysis. And it turned out that the native tissue actually has about 20% solid, 80% water. The ECM has about 94% water, 6% solids. And the seeded scaffold goes back towards the native state. You can see it here. It basically decreases the water content. And part of it, we think, is the masking of those glycosaminoglycan. Another part is the fact that just basically occupy some space and don't allow water to penetrate in that area. And then last thing we did was to try and measure if we have any phase changes on the bulk using a DSC, so the differential scanning calorimetry. And what we saw is that the ECM has a uni-peak. This is the black line over here. It has one peak, which is around the melting temperature of collagen, of pure collagen. But then when we look at the native tissue, we have also this small peak of ECM. Relatively, it's only about 6% or 4% of the native tissue. But we also have this characteristic three-peak structure, which are characteristic for myosin, sarcomerican proteins, and actin denaturation. And, well, of course, the mesenchymal stem cells that we have seeded, they are not exactly, I would say, cardiomyocytes. So they don't have this entire structure in there. But they are capable of remodeling the ECM. And that's why when you look at the dotted line, which is quite difficult to see perhaps, the dotted line goes something like that behind the... So this dotted line shows us that there is some regaining of complexity in the structure that we have generated, or that the cells actually generated within. And at the first transition temperature, moving from this point to this point, though it is not particularly significant between the reseeded and the ECM, but there are some significant difference between the ECM and the native tissue over there. And so we think that if we will increase our studies and the repetition groups, we might be able to prove that this first transition point may be indicative of proper cell integration within the ECM. So I think I will finish up with this, and I've tried to prove a point here today that cells can influence the scaffold biophysical properties, and they can actually restore the biophysical properties of scaffolds towards a native state. And if we give them enough time and we choose the correct cells that are capable of doing it, at the long run we might be able to prepare implants better for transplantation than just implanting them directly, or devising materials in the lab and testing them in vivo all the time to see what happens. I mean, we can test them probably with cells prior to that and see how well cells react to them in the lab. It will save a lot of funds and efforts in the long run. So I think that was the point I was trying to make today. I would like to give a few acknowledgements to Professor Marcel Makhlouf, who is the head of the program, and also in charge, together with Professor Subhavan Katraman, on the work done in our thrust. This is the older program members, as you can see here, danced together next to our entry door, and this is our thrust, in particular those two ladies in there, Dr. Udi Sarig, who happens to be my wife, but also a colleague and a brilliant scientist on her own. And she was supervising our PhD student, Gigi, over here in that work, most of the results that I was presenting today. And with that, I would like to thank you for staying up until this late time. And if you have any questions or discussion, I would appreciate that. Thank you. Yeah. Thank you.
In your study, you are taking a scaffold and seeding cells in it, and you're gaining biomechanical better results with no cell, only scaffold biomechanical properties will enhance with cell ingrowth. Mm-hmm. But these cells will be metabolically active. Will you get angiogenesis on these scaffolds? Will it be possible? These myocardial cells will be metabolically active. And if there won't be an angiogenesis, there won't be a functional capacity.
You're talking, let me just see if I got your question right. You're asking whether when we implant this into the body, whether we will see angiogenesis occurring inside, or do we have to add additional modifications to induce angiogenesis? This is an excellent question. I did not have the time to show this today. I have the slide, but I don't think time will allow us to show this. First of all, this material is very bioinductive. And when we implant it into animal models, we see an increase in angiogenesis on the first 14 days, which is much higher, about 2 or 3 folds higher than what you would see in the surrounding infarcts next to underlying infarcts, because we implanted as a patch material on a rat infarction model. And so angiogenesis, to answer your question simply, angiogenesis does occur in there. There is a lot of cell penetration. Actually, this ECM, when it is being degraded, so that the first cells that it meets when you implant it are macrophages. And what happens is that they change the phenotype of the macrophages for a favorable M2 over M1 ratio, if you're familiar with that. Meaning that the macrophages start dealing with this material as an acceptable material and they start doing constructive remodeling. During that process, they degrade the ECM. and they release a lot of growth factors that are present within the ECM, and matrikines, which are cryptic peptides of ECM. And when those are being released, they attract progenitors, which include hematopoietic progenitors, but also muscle progenitors. And I think we were seeing perhaps one of the first times that we are going to report soon, hopefully, about a process where we have actual cardiac-like or muscle, I wouldn't say cardiac, but it's a muscle-like phenotype that is being formed within a completely acellular material that was implanted on the cardiac tissue. So we do see angiogenesis occurring, we do see recruitment of progenitors and muscle-like being formed. We stand for specific myosin light chain and troponin I, as well as for a number of progenitor markers like GATA4 and c-KIT, and we see those being recruited into this material, whether this is injected or delivered as a patch in both situations. So angiogenesis does occur, and the cells that penetrate into the scaffold, also in vivo, they actually, as time progresses, they improve the biophysical properties of it to make it more matching to the heart. So it's actually quite a nice mechanism. But I think that the ability to study this, because the overall aim is not to use ECM. The overall aim is to develop some sort of easily robust and easily manufactured scaffold that could be used. But I think that in the process of designing, we need to go take one step backwards and look what happens in nature, what the cells are looking into the scaffold, what natural scaffolds can achieve and do, and then try to use that in our design processes.